Hi everybody, it's Mr. B here, and I am here today to talk to you guys about the process of photosynthesis. We're going to look at specifically what it is, how we can observe it, and where it can be found. And the way we're going to do this is through the observations of a common aquatic plant lab that measures the total amount of CO2 found in the water after respiration and photosynthesis occurs. But before we can get to that, we need to go over specifically what is photosynthesis. It is a biochemical process in which plants absorb light energy in order to convert it into a usable energy resource. That energy resource normally is ATP. So this process specifically occurs in the chloroplast, which again is a plant-only organelle. And the enzyme responsible for this breakdown is chlorophyll. So what happens is the chlorophyll traps the light energy within the chloroplast and then proceeds to break it down. So the overall components found within this reaction are carbon dioxide, oxygen, glucose, water, and light energy. If you take a look at this picture on the bottom right, you can see you have sunlight, water, and CO2 going into the plant, and the plant is producing oxygen and glucose. So specifically where this process gets housed is within the chloroplast. So again, the chloroplasts are found in the cells, and here's a nice little diagram of what a chloroplast looks like. So within the chloroplast are things called thycloids. And those thycloids are sac-like membranes that are actually responsible for housing the light energy and where this breakdown process occurs. So the chemical equation for photosynthesis is carbon dioxide plus water with the appearance and use of chlorophyll and sunlight or light energy yields to give you glucose or sugar and oxygen. So specifically, the way we're going to study this process is through a lab. Again, this lab is going to study the photosynthetic activity of aquatic plants under various amounts of light. So we're going to manipulate a photosynthetic production process by using aquatic plants and putting them under different amounts of light over a 24-hour period. The plan is to look indirectly at the release or emission of carbon dioxide into the water as a result of cellular respiration. And the reason why it's an indirect process is because cellular respiration is an ongoing process that all living things need to happen in order to survive. And as a result of cellular respiration, CO2 is a product. So this CO2 is then being admitted back into the water. And what essentially happens is that respiration cannot occur without glucose, and that glucose is produced through photosynthesis. So in order for respiration to produce CO2, photosynthesis has to occur and produce glucose. So the way in which we're going to measure the total amount of CO2 that's found in the water is through a chemical known as brothymal blue. And what it does is it specifically measures the total amount of CO2 found in water and then changes the color based on a pH scale. So if there is no carbon dioxide in the water, the solution will look blue, as here in the bottom left. If there is carbon dioxide present in that water and brothymal blue is added, that solution will change from a blue to a yellow or green, like this picture on the left. So both of these examples I've shown are actually the control groups that I used in my experiment. So again, the way in which we plan to measure the total amount of CO2 found in the solution is along the pH scale. And the way we're going to do that is specifically between the pHs of 6 and 7.5, because as photosynthesis occurs, the color should change in between this yellow and blue parameters. So for today's lab, we're going to test 4 aquatic plant samples in 4 test tubes. And what we're going to do is one sample is going to receive constant light over 24 hours. Another sample is going to receive a manipulated amount of light, which will be 50%. And then one sample will receive no light. And then we will also have a sample containing the carbon dioxide rich solution in which CO2 is already present. So the overall process of this lab is again to look to see the changes in color of the solutions over a 24-hour period of time. And then we're going to look to justify our results based on the color change in each sample. And we will identify which sample underwent the most photosynthesis, which one underwent the least amount of photosynthesis, and finally come up with some conclusions for this lab experiment. So the materials, here's just a picture of them. There was a variety of test tubes that were labeled specifically to show which ones received which light. There was distilled water, the 4 aquatic plant samples, aluminum foil, and brothymal blue. And again, these are the materials listed. So the procedure is the aquatic plants will be cut into 3 equal sections. All the sections will be the same length. That BTB control group, with and without carbon dioxide present, will be created and shown. Then the 4 test tubes will be labeled, showing the total amount of light each plant will receive. Once the controls are made, 30 milliliters of BTB is added to the 4 test tubes. Those 4 test tubes are 2/3 the way full with distilled water. And once you have the BTB added into the water, you can then proceed to place the plants into the solution. So again, the solution will sit under a 40-watt lamp for 24 hours, and 2 of the test tubes will be covered in tinfoil to manipulate the amount of light. So like I said, one will be completely covered, one will be 50% covered, and then we will have 2 solutions that are receiving 24 hours of constant light. Hey guys, this is Mr. B, and I am here today to go over with you guys the results from the aquatic plant lab after the 24-hour period in which it ran. So the first thing we need to do is go over the control groups again, and this one was the one in which BTB was added to H2O, and there was no CO2 available, so notice that it has this blue coloring to it. The next control was the one in which it was BTB, H2O, and we added CO2 through the addition of air through a straw, and notice that the color changed from blue to yellow. So now we're going to go over our results, and the first one we're going to look at is the plant sample that received 0 light, and notice in this one that the color still is that original blue, and it can be said that there was no photosynthesis that occurred because there was no light energy available. The next sample that we're going to look at is the one that received 50% light, and notice in this sample there was a bit of a color change. That color started going from blue to yellow, but it wasn't as significant yellow as we saw in the control, and again the reason is because this sample only received 50% light. The third sample that we're going to look at is the one that received 100% light, and notice that this sample is the closest one to the control, and the reason is because under the light, this reaction was able to continuously occur and produce CO2 that was added. So again, this sample went from the original blue color of the control to the yellow color. And the final sample we're going to look at is the one in which we added the already tampered with BTB solution in which CO2 was added, and notice that there was no color change. And the reason for this is because since CO2 was already added into the solution, the molecules were already not at equilibrium, so there was not any oxygen available for the experiment to run. So again, the one that experienced the most photosynthesis was the one that received 100% light. And the reason was because that light energy drove photosynthesis to produce glucose,
which ultimately then had the cells perform respiration, producing CO2 as a waste product.